New method of radical organism rejuvenation and significant prolongation of mammals and human life. Prolongation of life is the problem for all times and for all nations. In evolution, this problem concerns humans only. Realizing immortality of animated nature via reproduction of mortal organisms and having instincts of survival and self-preservation, humans always tried to resolve this contradiction of life and death. At all epochs, humanity showed a great interest in means of youth and health preservation and achieving of extra longevity. Such means were sought in ancient China, India, Greece, Egypt, by great civilizations of Central and South America. Traces of this search are lost in ages and belong to ancient history. The oldest Chinese medical handwritten book, Ne Qi Ne, contains many articles about aging. We can find descriptions of rejuvenation and immortality means in manuscripts of medieval-era alchemists. Facts of human extra-long life, which are mentioned in ancient books, seem to be exaggerated, because archaeological data proves that ancient people lived no longer than modern people. Species limit of lifetime was not overcome. Since the end of 19th century, systematic studies of human and mammals' life prolongation were started. For 100 years, scientists gathered a vast material about biology of aging. Many studies describe solving of gerontology's main objective, life prolongation. These studies contain information about how various factors affect lifetime of humans and animals. The most important contribution to increasing of some countries' population lifetime was done by social factors, environmental conditions, and achievements of medicine, which are not related to aging deceleration as such. These factors allowed to get closely to biological limits of lifetime, which are almost reached in Japan and in some of developed countries. Application of geriatric medications is targeted to prophylaxis of many diseases accompanying aging. Some scientists believe that extremely expensive measures aimed at curing main diseases of elderly people may add 10 years of life approximately. Thus, humanity's almost exhausted potentialities of lifetime increasing by traditional medical means, and now comes ahead a question of development of means and methods of radical affection on aging process itself. Many well-known medications increase average lifetime of human and animals. At the same time, maximum lifetime is not increased, which means that these medications are targeted on correction of aging pathological consequences, but not on fundamental processes of aging itself. In many life prolongation experiments, scientists used various lines of short-living laboratory animals with well-known inherently proven diseases. If a medication was efficient for treatment of these diseases, then average lifetime of animals increased, not affecting process of aging. Other methods evoke signs of human and animals' rejuvenation. At the same time, maximum lifetime is also increases. Expert analysis of modern situation and prospects of gerontological research's development presented by world's 49 leading gerontologists at the beginning of 2016 showed that majority of experts believe that a possibility of human maximum lifetime increasing is a solvable and near future problem. Nowadays, various techniques, methods, and systems of lifetime increasing are being studied. These techniques and methods can be divided into two groups based on aging process affection efficiency. Those increasing both average lifetime and maximum lifetime, and those increasing only average lifetime. The first group is of greater interest to us. Applied methods are of various natures, physical, chemical, and others. Here we examine known, but as a practice shows, less efficient means and methods of rejuvenation. Antioxidants. Application of antioxidants is based on the ability to bind free radicals, reaction compounds with unpaired electron, which appear during metabolism process, amount of which increases with age. Harman's hypothesis. These radicals may damage organisms' metabolism by affecting high molecular compounds of chromatin, DNA, RNA, collagen, and other proteins, thus causing cell membranes lipids peroxidation. Mammals have strong antioxidant system regulation, free radicals activity. However, efficiency of this system decreases with age. The most efficient antioxidant, mercaptoethylamine, increased average lifetime by 26%. Butyl hydroxytoluene by 45%, mainly because of tumor development deceleration. Maximum lifetime of animals was not increased. Then above-mentioned antioxidants were given to mice of long-living and diseases-resistant lines. No life prolongation was observed. Thus, the conclusion is the following: antioxidants do not decelerate aging process. Analysis of information gathered from antioxidant studies makes possible a conclusion that it is not possible to achieve significant aging deceleration and increasing of maximum lifetime when using antioxidants. However, many of them are official preparations and deficient at many diseases and necessary for normal vital functioning of an organism and health strengthening. That is why application of antioxidants is advised for increasing of human average lifetime. Adaptogens. According to Bjorksten's hypothesis, process of aging is related to development of linkages in DNA molecules, collagen, and other proteins by covalent and hydrogen links. Bjorksten signed out bacterial enzyme destroying these linkages. Its introduction to mice resulted in average lifetime increasing, which, according to the author, decelerated aging. After that, a group of adaptogens was discovered, which prevented development of linkages in particular molecules of conjunctive tissue collagen, penicillamine, semicarbazide, aminopropionitrile, and others. Now, in experiments on rats, mice, and other animals, it is proven that adaptogens, while inhibiting development of cross-linkages, almost do not affect maximum lifetime of animals. Adaptogens. These are tonic medications which increase adaptive properties and resistance of an organism to various factors, to provide above-mentioned effects, and to decelerate age-related changes. Mostly the following tinctures and herbal extracts are used: ginseng, eleutherococcus, flaxseed, devil's club, luzia, aralia, and pentocrine. However, applications of these preparations is limited because major part of adaptogens is counterindicated at essential hypertension and other age-related diseases. Exaggerated attitude towards these and some other herbs, euphorbia, Adam's rhododendron, Gastrodia elata and others, as to elixir of life, is often explained by cosmetic effect and stimulation influence to some processes in an organism. Direct experiments prove that only minor part of adaptogens, extracts of ginseng, eleutherococcus, and licorice root increased average lifetime of rats by less than 10%, almost without any effect to maximum lifetime of animals. Biostimulants. Biostimulants group includes a line of cytotoxic serums, antireticular, anti-ovary, antitesticular, and others, which are used in geriatrics. The most known aging prophylactic preparation is antireticular serum created by S. Schwartler in 1943, who linked aging process with changes in conjunctive tissues. Antireticular serum has a positive effect to conjunctive tissue system. However, there are no conclusive facts that antireticular serum affects human lifetime. Immunotropic preparations. Application of these preparations and techniques in order to prevent aging is based on hypothesis linking aging process with age-related changes of immune system, and first of all with thymus involution in humans and animals. There is information that hormonal matter of thymus humoral hormone, thymopoietin, thymosin, and others, and interleukin-2, isoprinosine, immunocytal, azathioprine, immunosuppressant, thymex, and thymolin, thymus extracts, and others decelerate thymus involution, cause normalizing effect to functions of various systems, prevent tumors development, stimulate protective properties of organism, but do not cause any marked effect to lifetime. Hormones. Studies of hormones' effect to lifetime are based on important role of neuroendocrine regulation mechanisms in the process of aging. Nowadays, it is proven that introduction of testosterone decreases lifetime of females and especially males of mammals. Minor life prolongation of animals was observed after injection of estradiol, oxytocin, vasopressin, and glucocorticoids. It is noted that large doses of dihydroepiandrosterone injected to mice caused rejuvenating effect. However, large doses of this hormone injected to humans cause side effects. Preparations affecting genetic system. In worldwide gerontological scientific literature, about half of all works are based on studying of aging genetics. In order to delay realization of genetic program and to decrease synthesis of proteins, possible initiators of aging, a number of transcription and translation inhibitors were tested: actinomycin D, olivomycin, cycloheximide, and others. The greatest effect was achieved after injection of olivomycin to rats. Average lifetime increased by 15.4%. Any attempts to stimulate activity of the aging genome with DNA and RNA preparations did not show significant result in life prolongation. Vitamins and microelements. In geriatrics, vitamins and vitamins complexes are used for deceleration of age-related changes. Effect of vitamins to lifetime was studied in experiments. Vitamin E did not affect lifetime of mice of various lines. Sakamoto. In experiments on mice carried out by Massey, effect of large doses of vitamin C resulted in increasing of average lifetime by 10%. However, maximum lifetime did not change. The author makes a conclusion that vitamin C cannot prolong human life significantly. Limited nutrition. Osborne carried out first systematic experiments on mammals. Experiments on rats proved that calories limited by 20–30% adequate diet resulted in prolongation of animals' life. In 1930–1943, McKay's scientific group carried out intensive studies, repeated afterwards by Ross, Barrows, Freeman, and Everett. Results of some experiments describe lifetime increase by 20–40% under effect of various conditions of nutrition limitations and even excluding from diet of only one of amino acids, tryptophan. The greatest life prolongation results were achieved in experiments which was started from infancy. However, limitation of nutrition in middle and old ages was inefficient. Acupuncture. Age-related desynchronization of biorhythms causes acupuncture points' electrical activity decreasing. It is possible to normalize biorhythms functioning by affecting acupuncture points and channels of a human. It is known that when some acupuncture points are affected, energy of an organism significantly improves. Zhusan Li point is considered the point curing 100 diseases. Influence on this point causes normalizing, regulating, and tonic effect to an organism. According to traditional Chinese medicine, above-mentioned point is used for life prolongation. There is health-improving system according to which biologically active areas are massaged by influencing acupuncture point E36, Zhusan Li. Similar influence activates hypothalamus reticulated formation amygdalaris complex system. As a result of such activation in humans, the following signs were noted: age-related changes deceleration, improvement of cell and tissue metabolism, and immune reactivity. There is no data about effect of this method to maximum lifetime. Physical factors are more efficient in rejuvenation of an organism. Electromagnetic fields. It was found that after mice exposure to negative charge low-voltage electrostatic field, average lifetime of mice increased by 12%. Turnbull studied effect of electrostatic field voltage 15 kV/m on male mice of CBA inbred line. In comparison with lifetime of animals from screening group, average lifetime of experimental animals in negative charge field was longer by 11%. Maximum lifetime increased by 13%, but mice of CBA line have high probability of tumors development. In this experiment, maximum lifetime did not exceed specific limits. The result was achieved thanks to decreasing of tumor's development frequency after exposure to electrostatic field. Starting from 1939 and until our days, scientists have carried out a number of experimental researches showing influence of pulse magnetic fields of 4 to 7 Hz frequency on lifetime and survival rates of various types of animals. Experiments carried out by Van Dijk and Halpern showed a decreasing of mice and rats' lifetime when they were influenced by a north magnetic pole. At the same time, it was found that south magnetic field has stimulating and rejuvenating effects to all living matter. Average lifetime of mice was 38% higher, and maximum lifetime was 40% higher than of mice in a control group. At that, high-frequency pulse magnetic field of more than 20 GHz frequency and 100 millitesla density had much more evident effect to life duration of mice and monkeys. Becker. Application of torsion fields for rejuvenation of mammals and humans is a comparatively new and efficient method. Torsion field had a greater effect in experiments carried out by Schmidt. Effect of right-hand polarized torsion field of 40 GHz frequency was administered to old rats, 24-27 months. Objective of experiments was to increase adaptive reaction of activation in mammals. In 1-2 weeks after beginning of exposure to torsion field, animals showed first signs of rejuvenation. After 4 or 5 months, animals could be distinguished from younger ones only by size of bodies. They were more active. Thin yellow hardened hair was replaced by thick white, thick and soft hair. Yellowish eye sclera became bright pink. Skin changed from rough and thick to soft and flexible. Thymus increased. Sexual cycle normalized. This effect was strong and prolonged. A number of scientists, MacLean, Walters, Hubbard, proved that right-hand polarized torsion field promoted significant increase, in some cases by several times, of average lifetime of mice, rats and other animals. At the same time, left-hand polarized field had damaging effect. These experiments present a great interest for studying of torsion field effect as a way to prolong human life. We have performed a study of torsion field effect on 12 women volunteers, aged 51-58. All women were at menopause period. In all of them, complete stopping of monthly cycle have been registered for more than 2 years. To apply modulated torsion field, we used an experimental device, Metatron Z100, with a high-frequency torsion generator with field generation frequency of 100 GHz and a possibility to generate low-frequency 250 Hz micropulse modulation of a signal and influence organism with right and left-hand polarized fields, according to protector-destructor principle. Throughout the experiment, 12 months, organisms of women were subjected to effect of right-hand polarized torsion field, once in a week with 10 minutes exposition. In 5-8 months after beginning of torsion field exposure, the first signs of rejuvenation appeared in women. Subjectively, all women noted improved well-being, memory, absence of depressions, decreasing of nervousness, improved sleep. Objectively, in all women, estrogen level increased by 2.2 times in comparison with the initial data. In all women, without exception, we registered increased muscle tone, improved skin elasticity, decreasing of sagginess, and smoothing of wrinkles. 9 women also noted improvement of eyesight. 7 women noted complete disappearance of pain sensations along spine and in lumbar area during walking and standing, joints pain, which were registered in them previously. In 6 women, monthly cycle restored. In all 6 of them, we registered a complete multi-stage sexual cycle with restoration of sequence and continuation of stages. Generalization of acquired data allowed to acknowledge evident bioadaptive and geroprotective effect of high-frequency modulated torsion field. Its non-specific mechanisms of influence to female organism was registered by studying of a combined interrelated blood indices, including increasing of erythrocytes number and decreasing normalization of a general number of leukocytes, increasing of immunocompetent cells contents. Using multi-component cellular contents of blood and calculation indices of reactivity and intoxication, we also managed register reconstruction of statutory indicators of protective and detoxing processes in women after exposure to torsion field. Surely, endocrine regulation is the most ancient form of control, and at aging, it undergoes significant involution. Due to this, complete restoration of hypothalamic gonadotrophic regulation in aged females after exposure to a torsion field is a significant result. Therefore, we managed not only to achieve metabolic and external rejuvenation of women, but also to restore reproductive function. After completion of our studies, one woman, 54 years old, got pregnant and gave birth to a healthy child. Therefore, given analysis of approaches, methods, and techniques of life prolongation allows to note effective torsion field to human's organism as one of the most promising for human and requiring further deep studies.